Welcome back to another edition of MIP 302. Today we're going to be talking about exercise 10, which if you haven't read yet is about bacterial genetics. Specifically, we'll be talking about how bacteria exchange genetic material amongst themselves, and these are the three ways that are depicted in your lab manual. So we will be talking about transformation, conjugation, transduction. Here's a little image of some bacteria. And once again, after this video, I will attach some cute pictures of my dog, Tucker, when he was a baby. So, fun times. All right, let's jump in. So transformation is when a bacteria finds some DNA, some free-floating naked DNA, in its environment and uptakes it. This DNA will have been from a bacterial cell that died and lysed, so broke open, and released this DNA into the environment. So another bacterial cell comes along, finds it, and reincorporates it into its DNA. This could be like a little plasmid or something, right? Absorb it, take it in. So what I would highly suggest to try and remember these differences between these three different ways is trying to come up with a mnemonic for yourself that you can remember that'll help you remember what these things stand for. So I think that the environment transforms all the time. It's always changing. It's very dynamic. So that'll help me remember that this DNA is uptaken from the environment because the environment is transformational. It's really cool. Okay, something like that. Okay, conjugation. So this lab that we would have done in class, but now you're just going to get the results for, is dealing with this concept. So I would really understand this part of the reading for sure. Essentially, conjugation is direct cell-to-cell contact between bacterial cells. And with that direct cell-to-cell contact, there is a donor bacteria and a recipient bacteria, F- and F+. So this bacteria here would have a plasmid or a fertility factor that has the ability, the gene within it, to create a pilus for these cells to mate. And because it has that gene, once it gets in contact with another bacteria, it can transfer that plasmid into the recipient or the F- factor. Once that plasmid is in that recipient cell, it would now be able to form a sex pilus with another cell that it would interact with because it would have a copy, or it would have that plasmid as well, right? So on that plasmid, there's a gene that allows this sort of thing to happen. All right, we'll come back to this concept when we talk about the lab. All right, now let's talk about transduction. Transduction, the main thing you need to remember is that it requires a bacteriophage. So a bacteriophage, I've drawn one here. Sometimes can look something like this. It's a rudimentary drawing. I'm not a good artist, but essentially it's a phage, or just like from our phage lab that we had done earlier, that will replicate inside of a bacteria. So there are two methods that your lab manual describes here: generalized transduction, where during the viral replication part of that phase, a bacterial genome, part of that genome, is accidentally incorporated. So here's a picture, because I think pictures are 1,000 words, of what that looks like. That might help you visualize it better. All right, the other one is specialized transduction that your book talks about, where an error is made when a lysogenic phage excises a portion of bacterial DNA. So here's a picture that kind of describes that process. Like I said, I feel like pictures explain a lot better than sometimes words do. So here's what that looks like. So what are we doing in this particular lab? We are looking at conjugation, and the way we're doing this is through two different E. coli strains. One is HT99, which has resistance to this particular antibiotic here, and is our F+, which means that on that plasmid it has the gene to create a pilus. The other strain that we're looking at is J53R, which has resistance to this particular antibiotic and is our recipient. So one of the things we would do is we want to look at how these different individual strains perform on different TSA plates with either this antibiotic, that antibiotic, or both antibiotics. And if we were to mate these for a certain time, these two strains, what would be the outcome of that on a plate that has both of these antibiotics? So we would add 0.1 ml of the HT99 E. coli strain to the J53R strain, 0.9 ml, into 5 ml of TSB and incubate that for 3 to 24 hours. Meanwhile, we would take those individual strains and put them on plates. We put them on a plate with just this particular antibiotic. We put them on a plate with just that antibiotic. And then we would put them on a plate that has both different antibiotics. And we would add 1 drop with our pipetter onto each and then spread them out. And, well, let's think about what would happen. Would we expect to see growth on, let's say, this particular plate? And if so, which one would grow? Well, the one with the resistance to that antibiotic would grow. Same thing for the other ones. So think about what antibiotic is on there, and if it's both, does it have resistance to one antibiotic or does it have resistance to both antibiotics? And if they've mated, what genes have they shared, right? So has it shared the gene for antibiotic resistance? Well, if they've made it, hopefully so. So hopefully thinking about it this way and seeing these pictures will help you in terms of answering your questions. I also have a few tips that can help answer some questions that I commonly see students have a problem with in the past. So this is a few different things that I've come across in the past that students often have a hard time with on the post lab, and these are just a few tips on how you can start to think about those questions. Particularly, let's start with question number 3 on your post lab. And when you're thinking about this question in particular, think about what equation do we use to determine concentration in the original stock. It's the only equation, hint, hint, that you've learned this whole time. And also try and think about, did we perform a dilution on our mated broth tube? Once we had it made. So that should help you with that question, hopefully. And question number 5. So question number 5 is pretty long. Just a few little hints on that. If you can see something on a microscope with your 100x oil immersion lens, then it can't fit through a 0.2 micron filter. Anything you can, it's just a good rule of thumb to think about. It's hard to know, and I don't expect you all to know sizes of things, but this is just a good rule of thumb if you're ever questioning what can actually fit through that small of a filter. The other thing that some people might not know, DNase is an enzyme that degrades free-floating DNA. So any DNA that it's in contact with, DNase will degrade. All right, so hopefully that'll help you. As promised, here are those pictures of my dog.